Hello. Okay. Thanks everyone for coming. It's my pleasure to present Dr. Sze Chai Kwok, who's an associate professor of cognitive neuroscience at the Division of Natural and Applied Sciences at the Duke Kunshan University in Shanghai, China. Thank you, Sze Chai, for coming.
Hello everybody. Thank you, Ed. Thanks so much. Well, very impressed this lecture hall and so thanks for the introduction and all the hospitality. I have had some time visiting some of the lab, but I will continue the visit after the talk. So my name is Sze Chai Kwok. I'm now at Duke Kunshan University, and then I'll show you where it is. So it's a bit far to come here. So it's China, and then this is Shanghai, and this is right next to Shanghai. This is a place, a city called Kunshan. So it's about 1 hour by car to go to Kunshan, and this is where I'm at. This is a new university, and it's been around for only 10 years. And it's a joint venture with a local Chinese university called Wuhan and Duke in America. And this is a liberal arts college. It's very, very beautiful and stuff, but it's new. But for the work that I'm going to talk to you today, and for the research, it was conducted when I was with East China Normal University, which is one of the top 4 in Shanghai. And this is where I still run my lab, my non-human primate laboratory, as well where I have my research group, like postdocs, graduate students, and so forth and so on. I also hold an adjunct position with them. For the next few slides, I'm going to show you what the laboratory have been doing in the non-human primate stuff. So I borrowed some slides made by my students. You can see here. Let me see if that works. Works here. Works here. It works. So here is a new setup where we have this eye checker. And we're doing like VAFC, these animals that you can see from here doing a memory recognition task. Apart from this, we're also building a new, on the top right, we're building a new rewarding system. Instead, we're going to use smoothies. We blend the food with milk and stuff instead of water, so we can hopefully make them more motivated without water restriction. This is a model that we're learning from the Oxford colleagues. Apart from this, we also have, this is the pictures where we have the surgeries. I think I saw the TMS at laboratory as well. And then we also have like a TMS experiment, like transcranial magnetic stimulation, that is the monkey's fixation. And this is the navigator. And they were doing a metacognitive task. So here we studied their performance as well as their confidence. That we are not going to talk about today, but then we have a new paper in communications biology, basically showing how people are, how monkeys are capable of introspective metacognition, as well as the role of lateral prefrontal cortex that we do. Let me see. Here is the setup where we do the electrophysiology, as well as the task. So this is, the main question that we're doing is on memory retrieval, and we train monkeys on the temporal order judgment memory task. This is a paper we've got from this in the life. Basically, we show monkeys are able to, there's some evidence behaviorally of replay, of how they replay the video clip they have seen when they're performing memory judgment. And together with that, in further step with that, we also have the recording. You can hear the sound of the spike. And together with this temporal order judgment task, we're looking into the area in the posterior parietal cortex and how these neurons may work together to track the passage of time, as well as how they synchronize to support temporal order memory. I wish I could talk more on that, but I didn't prepare to talk on this one, but this is something ongoing that we're looking into. Okay, now, so in order for us to study episodic memory, I think the the first step, the first thing we need to do is to understand what an episode is. Then this is how we can get us started. So let's say we look into this video, either monkeys watching it or we watching it. It looks simple. I think I don't need this one. If you look at that, it's a simple naturalistic video, but then it's actually consisting of a lot of information. You see animals, objects moving, they're interacting. It's different color, and the movie is shot differently with the camera moving and so forth and so on. Like there's a boundary there if you notice, and then other monkeys come in, all this stuff. So this is the episode. We try to use video or naturalistic videos to mimic how we process a video. Then to study that is not that trivial. I'm going in this talk. There are two concepts that I hope I would bring them to you. First of all, is we want to get away from the traditional sense of simple condition with activation or firing correspondence. Rather, we're going to look into this mixed selectivity concept. And second thing is like, apart from the neuronal activity that I will show you, we want to see whether we can study the whole brain in a more global, holistic way. And these are the two concepts that would sort of tie my talk. The first concept is here, like what I just said about, when you look into episode, there's so many stuff there, like stimuli, color, action, movements, animal, and so forth and so on. It's not easy to tackle with the traditional understanding of neurons, right? Previously, in the past, neurons is conceived as simple, specialized, as in if you show them one kind of stimuli, then you expect one kind of activation. If you show them another kind of stimulus, then you expect another kind of activation. But then apparently it doesn't really work. And about 10 years ago, by a group of people, including Earl Muller and colleagues, they started this idea, or championed this idea that there's mixed selectivity in our neuronal system. So the idea, I can illustrate that here. So let's say, if you look at hypothesis one, a number of population of neurons, like three kinds, blue, yellow, different neurons there. And let's say some features is represented by a kind of neuron, like big circle. The big blue circle is doing one kind of stimuli, feature one. And then, but they also respond to a combination of other features, like these neurons here, right, the feature 3 and 2. In this situation, these two kind of cells category can be identified. But on the right, if you look at the second hypothesis, this is what the idea of the mixed activity is a bit more complicated, right? This population is characterized by mixed activity for 3 different features. Like a few units are strictly, like the big red one here, it's still very selective for a specific feature, but then most of the others respond to a different combinations of features. In this case, it's not easy to categorize cell categories. Yeah, go. Yeah, sure. Can I?
It highly depends on the cortex you're recording from, right? Because if you go to primary cortices, they are extremely selective.
Yeah, that's good.
If you go to prefrontal cortex, for example,
Yeah.
you find mixed selectivity everywhere. So it highly depends where you're recording from.
Yeah, I think you're right. So far in the literature that I'll show you in the next slide, they found in the lateral parietal cortex, and for this paper, I think it's like a frontal cortex. In fact, frontal cortex. Yeah, prefrontal cortex. Now, there's some advantages. For example, it helps with the nonlinear integration of multiple inputs to encode information, but also to help decode downstream as well. Then, like what you just said, is particularly important for the higher cognition because it provides the higher dimensionality, whereas the lower visual cortex is presumably simpler, and then that traditional sense may still hold. Some examples. These are new review paper, I think it's Frontiers, and they also listed out a number of other examples with rodents, actually. But I only pick out those with primates. And you can see from here, all of them, in fact, across different tasks, still in the lateral intraparietal lobule. So it's still very like true that they found it here, and they found it in prefrontal cortex, and I'm going to show you those new evidence in another brain region, in the medial parietal area in my talk. But I think it's a good observation. So here you can see the different tasks, different conditions, and then just mix the activity. I think the concept is clear like that. And the project that I show you is actually done with a very simple paradigm. 3 seconds, and a break, and then another movie. And they watch, let's say on that day, each of these movies 30 times. We intermix the order, and they will just watch it like this. Okay, so they have 90 times of viewing each day, and then we record the activity using this device. It's built by made by Charlie Gray, and then the good thing is that they have multi-electrodes. The one we bought is only 32, and then it's conically implanted, and then we can independently move the electrodes as according your needs, so I can move it on a daily basis. So you can record different depth and different neurons across states. And this is the example that we did the recording from, where let's say we call them 2 of them, Mercury and Jupiter. The blue one is the cartoon showing where the device and this area, the angle that we are going into. We use a CT and MRI overlaying, but you can see the electrodes at the beginning of the experiments. And later on, we move the electrodes down there. No, that actually is the medial part. So this is the surgical plane. Is right in the back of the brain. As far as we know, I haven't seen any monkeys' electrophysiology showing neurons from this brain region. So it's a medial posterior part of the parietal cortex. The data I'm showing you today is coming from the dorsal, the surface, very dorsal part of it. Then the memory study that I showed you at the beginning, we're recording towards the deeper medial part of it. And this I never show where we do the recording. This the 2 monkeys, Jupiter, Mercury, and we have a 3rd monkey using a different way of doing the cells as well. So here's some example that show you on how the neurons are like. So let's say they're watching these 3 videos, right? And on this day, on that particular day, we got 17 units that we got, which is listed on the Y here, the unit 1 to 17. This is time, progression of the video, and you see that they are labeled in different color, but the same neurons, The neurons do respond differently. If you look at unit 16, 17, they're more active here than the other 2, right? Whereas other neurons also respond in a different way when they watch this. So when it comes to that situation, then it's become clear it's not easy really to have this single condition versus a single response pattern kind of idea. And we have to find a way to tackle that. And before going to do that, I'm going to just show you some standard thing that we did with the spike. So here, the first analysis is to compare the 3 content types, the 3 primate, non-primate, and scenery. They're labeled in 3 different colors, green, blue, and red. The y-axis here is the repetition. So each video was shown 30 times, and they watched 90 times for that day. And the 3 colors refer to the 3 color response. So here, let's say we pick this neuron. You can see it is responding particularly strongly for the primate video than the other types, and pre and post referring to the before the start of the video and the end of the video as a control baseline condition, baseline. This is another neuron, so in this, responding slightly higher for scenery, of course, overall, and here is responding for for the non-primate thing. And there's some neurons that don't really, selective or responsive to any of the particular videos. Then for completeness, we also look into the other possibility. So this neuron is not responding so strongly with primates, and this one is not strongly with non-primate, and so forth and so on. So each of the dot here referring to spike, so the action potential across time per trial for that neuron. Okay. We also have a summary. From this study, we have got 375. And if you look at the outer circle here, this yellow one should be red, the scenery. This listing the number of neurons that are responsive to each of the 3 video content types. Okay. In this experiment, we actually, because you remember we showed them repeatedly, so we also have this kind of repetition effect. So repetition effect is showing this, the inner circle, which I'm not going to talk about. So basically, if we show the same stimuli repeatedly, the neurons will decrease their firing rate, and this repetition suppression has been known in IT cortex and in fMRI studies as well. And we found the same thing with a quarter of the neurons. The more they see the same video, they decrease their firing. And then interestingly, we found another kind, which is called enhancement neurons. The more they watch the same video, they actually increase their firing rate. I found it very fascinating as well, as if the neurons are processing the video content and learn something out of it. So this is showing the inner circle. Now, back to our main question here. So what do we do with this complex video stimuli and the neuronal thing? So we borrowed this idea from a paper by Michael Clark, first of all is Adams, that came a couple of years ago. And they use this idea of anthogram. Anthogram is the way to index or measure animals' lateral behavior. They use that to analyze the video I use that to analyze a video. So if you look at this chart here, I can zoom in and show you what's like this video feature labeling. So essentially, this is time for video, but as a function of movie frame, we label each of the frames using this anthogram idea, using annotation. So some examples like where there's anything like foraging behavior, whether they're showing any genital of the maquette, how many animals in it, whether they're making eye contact, whether there are other actions. So on a frame by frame, we semi-automatically as well as manually went through all the video frames and label it. Then for all the 18 videos that we have used, then we can have a resolution of single frame of the events that have been shown for all the videos.
You also have the position of the monkey, the eye, the position of the eye.
So we recorded from 3 monkeys with neurons, and one of them is eye movement. But in addition, we also performed another behavioral experiment using 2 additional monkeys that we record eye movement as well. That I'll have a bit of analysis on eye movements.
As a follow-up to that, when the monkeys watch the same video again and again, do they put their attention on the same spot of the video repetitively, or do they look at new features in the same video?
It's a very good question. I mean, it's not going to one way or the other, so a mixture of both. They show some gaze consistency across the repetition, but it's also modulated by what is being shown. So I do have this analysis, I think, after the neuron stuff. It's a good question, both of them, yeah. And in fact, eye check eye movement is also important to help us understand the neuronal responses as well. But the idea is clear with that. In addition to this higher level sort of features, because action, aggression, grooming, face, genital, all this stuff, we also look into the 4 low-level features, just need to complete the story. For example, optic flow, the rest of it, we analyze the video using some established algorithm to look in optic flow, and then we have the so-called saturation, luminance, contrast, and so forth and so on. And here is 18 videos, and we just plotted out this on, again, frame by frame, moment by moment, with the 4 levels of stimuli features like luminance, contrast, and so forth and so on. This is showing the correlation. So together with the 52 empirical items, we have a list of 56 items for the analysis. Okay. So how do we do that? We have 56. We have the neuronal firing, and we resorted to a way called the lasso regression, is least absolute shrinkage and selection operator. So essentially, it's just a regression. It's kind of regression. So we use this. We can construct a lasso regression to model the modulation of the 52 items as well as the 4 low-level items as variables on the neuronal activity. So the simplest thing is that the model or the regression would penalize each of the lines referring the features. We start with 56 features, and when they come down to 0, they will just get removed. The mean square error is the way that we use. So we pick the point where it's lowest. In this case, for this neuron, it's going to 21. So 21 of features will stay here. The features or items are listed on the right here. You see, for example, in this case, we have camera zoom, count, phase, and so forth. And the X here referring to the coefficient. So how strongly each of features moderate the firing pattern. Okay. We also check using this selected set of features and predict the firing activity. So we train the model just to sanity check.
What is the bottom line? In the graph on the middle? Yeah.
The what?
The highest correlation coefficient is for what? I cannot read.
The coefficient here?
Yeah.
Oh, so this means that factor or the feature is moderating more strongly.
But what is the label? The label of the last row in the middle?
Here? No.
No, in the bottom. Yeah, that one. The highest correlation is for what? For what feature?
So this is called camera zooming out.
And the lowest, yeah.
The 0 one is the strike, foraging, good foraging. Allogram. Yeah. This is actually strong. It's not. Oh, it's not lower one. Just like have positive moderation, negative moderation. The labels. Can you not see here? Right. This camera zooming in, counts, ingesting food, allogram.
Ingesting foods. Okay.
Yeah, allogram. But this is only for that neuron. That is only for one single neuron. So we run this analysis using neuron activity for one neuron for all 18 videos to produce or to have the last 21 features that are important. So another, okay, I'm going to show you more clear here. So let's say we have this video. This is frame, and this is video frame, and so forth and so on. And panel B is just the raster plot I show you. Across time, the neuron is firing up and down. Each of the dots, again, is action potential. It's a spike, okay? So it's got this kind of thing. You can somehow see some segments, they're more firing higher, maybe because of the content there, possibly. But again, we don't know. So this, at the bottom, this is the result from the lasso. This is a number of features that are found to be moderating the firing rates for that video more significantly. And red and blue refer to either positive or negative moderation for the firing rates. So this is a way that we do for neuron per video to obtain this. Okay, so now we can repeat the same thing with all 375 neurons. Each of them we did one by one, and this is a summary of what we got. Now you're interested in the features. The features are listed here, 52 features, higher level like grooming, fighting, eating food, and so on, that you can see. It's on bioarchive. I didn't have time to show you the thing, but then I could show you the bioarchive there. And the bottom four are the low-level features like luminance and so forth. You can see they're actually very strong. Hang on. So this is the 8 categories. I labeled them 8 different colors, from purple, some kind of green, red, and so forth and so on. So we have things you can see. So this is aggression. This is grooming. This is foraging. This is a genital. This is the number of count of animals. This is a face visibility. And this is a camera zooming in, zooming out, panning, cutting, and so on. This is the lower level features. So there are 8 separate categories. And then all the features are listed here. I didn't realize that the words are too small on screen. And here is the 375 neurons organized by monkey Jupiter, monkey Mercury, and monkey Galen. Separate them of them. And the color here refers to coefficient, like I showed you earlier on, whether it's positive in red, negative in green, and white is like it's close to 0. It's not so significant. And you can see for each of the neurons, they do show a different kind of configuration of the coefficient in response to each of the features per neurons. And panel B here is showing the proportion of neurons that are responsive to each of these rows. For instance, there are like a high proportion of them for lower level features, and some of them are less interesting for the neurons. Some of them are consistent. And the line, the dashed line here, referring to chance level. So we run some other chi-square and permutations. Say like even by chance, we should expect at least 27 neurons out of this pool, and you can see most of them are showing like significant more neurons to the features. And in the final panel is a coefficient. So the gray label is the significant sort of coefficients across the group of neurons. So this is showing you like this number of neurons are responsive to different features, to pattern of different features, as if they're processing the episode in this way. And we did a couple of control analysis. So reviewers come back and say, look, have you considered, because the area we're recording is like a higher visual cortex in these words. So how about the neuronal transmission from visual cortex? Have you considered that? So we ran a couple of control analysis by shifting the firing rate with the video by 40 to 100 milliseconds, 40 milliseconds, 100 milliseconds. The result remain pretty much stable, the same, indicating this transmission is not a big issue. But it makes sense if you watch movie, right? Per frame is about 40 milliseconds, so and each frame pretty much the same, so that we shouldn't expect that much change. Another control analysis is back to your question earlier. What about eye movements? So we've been asked, say, look, are you sure this activity or the modulation is really coming from the features, but not by saccadic movement, eye movement? And how do you distinguish the contribution of the two? We can't, but then we looked into the data again. Now with this, one of the monkeys, Galen monkey, we have this same pattern, same thing. Now we analyze the saccade also on a frame by frame basis. So we see whether that frame is a saccade or no. And then we use that as an additional regressor variable, and we call it the saccade regressor. So it's the 57 regressor. Now after factoring this, the majority of results pretty much stay the same, even in fact that gets stronger statistically, probably because of the effect of eye movements being regressed out. But of course, eye movement is like modulating activity for a lot of neurons, despite the coefficient being relatively small. But this is the way that we try to account for eye movement per se. Please.
What about the effect of attention, especially with the eye fixation? Say, for example, your stimuli are very naturalistic, right? So there could be many things happening at once. And what if there's a different type of stimulus, and one of the trials the monkey is not paying attention to that, but is paying attention to this other thing. And in the next one, he's now paying attention to that other thing. So are these maps of correlations with features, do they dynamically change? Are they modulated by the shift of attention from the monkey?
Yeah, I think that's inevitable, right? Even for us, we have this kind of additional factors of the attention. The way we did the analysis, we used the mean firing rate across the 30 viewings, 30 repetitions. So in that case, the attention is not particularly control, right? Because we use the mean firing rate. So let's say some of the trials, some of they got absent-minded. That is possible, yeah. But here, yeah. So actually, it will be a good point for discussion how attention related to where they look and how consistently they look across the viewing. That is something we should consider. So another way we visualize the data, I understand it's probably small here, is we make a little, let's see if it works. I will show you this. I think it's quite pretty. Where are we? So I'll put it on the web as well. So this need to display. So each of the yellow dots are each of the neurons, of each of the 300 neurons. And if you click on the bigger disk referring to the categories, like this is the low-level features, foraging, genital, face visibility, count, grooming, aggression. So with the 8 categories, and if you click on, for example, here, you see aggression, and then there's the neurons that have been activated or modulated by them. And then if you click on like each of the individual neurons, you see how that neuron is modulated by what features and so forth and so on. This is like a camera, and the neurons are the same thing. So we can play around with that to give you a sense of how altogether these neurons are being modulated. I think it's quite a lovely display to do. I'm going to move this away. Move this away. Eh? Okay, now back here, so this is the way to summarize that, right? So the 8 colors refer to the 8 categories, and the 82 of the units are modulated by everything except the blue one, except grooming, I think. And then 63 of them are modulated by everything except the genital, and same thing, we did the same thing across time. And what you can see is like these neurons, on the right here as well, we just see by the 8 categories, the total number of neurons are modulated by them. So this is coming back to the original was about the mixed selectivity. So each neuron is not really just modulated by a single kind of features, single kind of features, but a set of features, as shown in this idea. Is that okay? Now, the next thing we did is like back to the eye movement now. We did have a collect some eye movement from 3 monkeys, so Galen and monkey K and monkey P. That these two did not get the neural recording. We used grid analysis. Imagine this is the monitor, the screen that we use, and we break it down into 144 grid cells, like different boxes. This is this example of the eye chase. And what we can now do is like we can analyze the viewing for each of the video kinds, and there are 3 dimensions here. So we have the frame from frame 1 to frame 750, and we have the grid, the grid being 0 to 144, all the grids, and the color, the heat map referring to the viewing count, or fixation count across the viewing. So here where you can see for that video where they look at what time and how repeated they look. Okay, this is just a visualization. Here, instead of using firing rate, we use the gaze consistency as a variable for the last regression. So because each time they look, right, but we can analyze how consistent using this method across the 30 viewings. And from here, you can also see some interesting pattern. So the 3 monkeys do show some similarity in terms of the consistency for certain categories of the feature, but also some differences. And again, the same thing, this is coefficiency. The only thing is that there's not neuron data here. It's only the viewing, the movement or watching pattern analysis for the thing here. So what we've done with the eye movement is that we regress our eye saccade effect, but here we also look into how they look across time. But it's still not tackling the second question by you about the attention. This is where they look, but the attention or the brain network and the attention system is not, fully elucidated here. So next couple of things I can go faster. So essentially, we used a liver node decoder, support vector machine to do decoding. We use single neuron firing pattern to see whether we can decode what video is that. So this neuron, 0056, can be used. This is firing rates. This is the decoding accuracy, the prediction accuracy. This is the permutation chance. So we train the decoder with 1 trial and then with 29 trials, and we test on the last trial and see whether we can decode which video that monkey is watching using the firing rate. We can do that, and it's quite successful on at least half of the neurons, where here we have about 155 neurons. On the blue, we call it vetted neurons that allow us to decode the 3D video type based on firing rates. Here, which just the mean is divided by the 3 video kinds. More interestingly, you can also see the decoding accuracy. Each of the dots referring to each of the 155 neurons. The decoding accuracy will also increase as a function of the number of selected features given by my earlier analysis, the lasso analysis. So that's showing that the features that the lasso will give us will also be related to how well we can decode, train a decoder. And it's particularly relevant for primary videos. I think it makes sense just because primary video is more interesting. The neurons should be containing more information compared to the scenery videos. Okay? So far, so good. So we talk about the episodic processing. Now, so far we talk about the content being a number of features, whatever in the scene. And it's only one aspect. The other aspect, if you're following, then apparently is what happens to progression of time. So about temporal elements. So not only this moment, presence, but also time. And this is another concept that I can walk you through. This is a concept called temporal receptive window. It's this analogy like spatial receptive window, that we have a temporal receptive window. And this is a set of nice studies by Yuri Hansen and Princeton. This is a review paper, but the idea is very, very nicely illustrated here. This is the human cortex, MRI data, labeled in 4 different colors. They claim, or they argue there's like hierarchy in terms of the way the brain regions will process time. Let me unpack a bit. Time, if you look into the X here, there are 5 different conditions. I put it here. So the first one is full story, then we have scrambled paragraph, scrambled sentences, scrambled words, and backward speech for the 5 conditions. And this is MRI study. What you will see more clearly is that in the red regions, they got lower or smaller temporal receptive regions like auditory cortex, visual cortex. The activity do not care about what you're showing them. Whether you listen to a full story or listening to a single word, that area activated in the same manner. But if you move up the hierarchy, and that's up to the angular gyrus, parietal cortex like precuneus, where we did the recording from my monkeys, and the medial parietal prefrontal cortex, they do care about it a lot. They can activate strongly only for like full story or like a big things. But if it's like moment, let's say a word or a sound, they don't get activated. That means these regions, at the high up at the apex of this hierarchy, they can hold information for a longer period of time to make sense. So analogy is like one single word means only solitude, but if you remember what I said over a sentence, a sequence, then you understand what I'm saying, right? And that applies not only speech, but also films, narrative, reading, auditory, and music. And there are a number of work that's been around, and then this idea has been supported not only with MRI, but also with ECOG, right? ECOG from patients recording, and so forth. So anyway, so this idea of how primary cortex is organized in terms of the processing of time, from short moment by moment processing to longer period of information. Then, because the area that we do recording for the monkeys is the precuneus, which I'm interested in, and then we try to see whether we can have this, apply this receptive window onto our neurons. So this is a decoding exercise, decoding results. Here is time bins for the 30 seconds of the movie. Here is the accuracy of the decoder for one particular neuron, 2-to-1 neuron. And if you look at the green thing here, this is the same exercise where I showed you earlier on. It's a live and out support vector machine. So we train with like 29 video trials and test on the last one to see whether we can guess which video that is. This is decoder. So what we show here is that if you use each of the time segment decoding, chance is about 33, 1/3, 40-ish, depends which permutation you're using. If you use the time bin, we have chance initially. But if you add up more of the epots to put in the decoder, we get an increase in the decoding accuracy, which is shown in green here. But if we do the same thing, but now using each of the epots independently, it doesn't increase in the decoding accuracy. We have a couple of control analysis. For example, on the right, we shuffle the label, and doesn't show this increase in the decoder accuracy across time for this neuron. And we did the same for every single neuron. So what it's showing here is that, look, this neuron, when it comes using decoder, it seems to be able to hold information across time, so that if we use more of the epochs of it, we use firing rate for the training. The firing rate, moment by moment firing rate, will increase, indicating this neuron may show some sort of longer temporal receptive windows, like what they show with the MRI studies as well. Here, the same neuron, but separating for the 3 video type, like primate, non-primate, and scenery, and then showing the same thing, but particularly strongly for primates. And here's the this is a summary. We found this pattern of increasing, accumulation of information for 57 of the neurons, and this is particularly strong for the primates. So about 1/6 of the neurons showing this sort of information accumulation across time. Then we get people ask, like, look, is that because you are would the result be moderated or affected by the number of epochs you choose? So instead of using 1 second time beam now, we use much smaller time beam, like per frame, 40 milliseconds per time frame, and we obtain pretty much the same result. In fact, a bit weaker here, even if we use more of the epochs for the training and testing. So the whole result holds. So this is the time aspect of the thing. And we talked to Mark Howard in Boston University, and he's got a good idea. He said, look, you separate analyze the content and the time. Can we do the same together? And he came up with this solution. We use the idea of optocorrelation. So optocorrelation is the representation of how the degree of similarity between one time series with his own lag version of the time series. Okay. So we just check how optocorrelate across time. Because now we have video content that provided by the angiogram, we have firing rates, then we could do that with the optocorrelation. This is one video example, and the black line here is the optocorrelation function for the video features, like the list of video features, a vector, and see how they're correlated across time. Okay, that's okay, only video. But now we can put into the firing rates. So each of the dots are referring to the different monkeys' firing rate. You can see the firing rate also, again, population-wise, would be showing this kind of modulation. And to some extent, you see the modulation is also optocorrelated following like how the videos is like. But here, not only having the video features for the optocorrelation, but we can show that across time. So you can see the firing rates, how it's modulated across time, follow to some extent of the video features. Okay. Then we did a number of tests. For example, instead of using, this is another video from Jupiter. We break, because there are 30 repetitions, we want to check how repetition effect may be present, right? So either we use all videos for all 30 repetitions, or we use the first 5 videos, first 5 repetition, first 15 repetition, and you to see they follow, also the optocorrelation is followed with the video, and then some of them are stronger when we use the first 5 trials, first 5 repetitions. That could well be due to what we said at the beginning. We have this repetition effect, either enhancement or depression or suppression. So it's getting more complicated with the repetition effect as well. But this idea you can show, we can see how the neurons will process or follow what's happening out there in both time and object and space. Okay. So a bit of time. So I will move on to the second part, but it will be brief, shorter. Any questions so far? So far we're looking into time and object, but only from a set of neurons acquired from here. So a tiny bit, 300, 400 neurons. It's interesting, but it doesn't really tell us how these neurons will communicate with the whole cortex or the whole brain when you do the processing, right? So I think it's not sufficient. And the other way people do, like in fact many of you do here, is neuroimaging, right? You can image monkeys when they watch the same movies, and then they can see what's happening in the whole brain, processing the episodes. And this, I picked out this paper because they claim they can use, in fact, only about 50% of the frames of the Bose signal frames would be informative enough for you to analyze. So basically, this idea is to use the whole brain thing. Then, can we combine the two sources of information, single neuron firing pattern with whole brain scanning? It's hard technically, right? You got monkeys in the scanner, it's already hard enough. So one solution, so this is the idea of how can we combine the two. One solution is as follows, that we can look into how each of the neurons firing rates with the Bose signal fluctuations across time during the episode processing. Then I noticed this paper by David Leopold, the first of his Park, and they published this in 2017 in Neuron. This is my own data, but what they did is a phage patch. They have some monkeys, they did the recording of the spike on their phage patch when they when they watch movies. But on a different set of monkeys, they show the same movie to them, but they put in a scanner and they acquire the whole brain activation across time. And what they did is as follows. So on top, you see they show the movie. This is neuron, whatever, the cell here. And this is a fluctuation of firing rates across different time points for this set of movies, okay? This is firing. And as I said, we can also acquire data from another group of monkeys watching the same movies, okay? And we did that, we acquired with Michael Oritz, who used to be in Newcastle in the UK, I think, but then I think the end of the lab now. So he helped us to show the same movies to two of his Newcastle English monkeys, and we acquired the data on the Bose signal. And this is two example voxels and how the Bose signal go up and down as a function of the time. Now, what we can do now is to look into the relationship between these neurons and the voxels. So I'll give you two examples. We can correlate this example cell, which is shown in one of the curve here, and with voxel 1. Voxel 1 is here. And you see they don't correlate really strongly, not very well. Understandably, maybe this voxel is not helping with the episode processing, okay? But if you choose another voxel, in this case, the one here, they correlate quite strongly, suggesting that neuron and that voxel activity somehow coincide or synchronize in some way during the experience of video watching. Now, we did that for each of the neurons, each of the 300 neurons. For each of the neurons, we correlate that with all the voxels in the whole brain. So for each neuron, then you can obtain each of these correlational maps. Either it's positive correlated or negative correlated. Then giving you a different level of information or picture of how firing rate of single neuron to whole brain processing. And then we have produced 300 maps, each map for each of the neurons. And this is let's say one of the matrix here is, each box of the matrix is the map. So it's 370 maps with 370 maps. This is we use a k-means algorithm to reduce to obtain the best sort of k value. Here is the result. Again, this is number of neurons by number of neurons. But then we organize it by the two monkeys here, and each of the neurons for all the neurons pattern. Now, do you see some sort of interesting some result coming out? This is the correlation, the reliability score of how two neurons will be grouped together based on the analysis of the correlational map. Now, we arranged all the cells now by video instead of by monkeys. You see there's some interesting pattern coming out. There's 6 different clusters coming out from this reliability results. Again, each of the squares, the correlation of 2 of the functional maps. It looks like when the functional maps would be different because they were naturally being classified into the partitioned into the different categories. This could be the experience number 1, experience number 2, video 1, video 2, video 3, video 4, and so forth and so on. It looks like when they have experienced a different episode or experience, the neurons would actually work differently together with the whole brain with this analysis. So this is the same result, just showing how the best value of K, how we do the clustering exercise. And on the right, each of the little maps referring to the neuron and Bow signal maps that I told you about, and there's 370 of them. The big ones here in the middle referring to the six episodes. Refer to the 6 episodes, 6 videos here. And the color referring to whether there's the neurons show like strong connectivity or positive connectivity or negative connectivity in the two colors. Okay, so it looks like when we look into using both information, we can display how they process an episode. In this case, the videos. Then another thing we could do with this result is that it looks nice with the middle is the result of the neuron and fMRI Bow signal maps that I showed you already. Then how do you compare with the other methods? What if we don't consider them together? Instead, on the right, we consider only the neuron, the cells, the same thing. Do we obtain the same results? It doesn't look that well, but it's actually some information there. For comparison, we can do the same thing, but now using only Bow signal, only fMRI frame to do the same thing. And then we compare how well each of the methods do. So here is a comparison. This is the neuron Bow signal map. This is only a neuronal map, and this is only the fMRI map. And this is the perfect ground truth, the ideal situation, because we hypothesize each of the videos would give us a separate experience, right? The 6 experience. And then we compare how well they fit together, and apparently you see the fitting is the best when we make use of both neuron and functional information, functional MRI information. And then to compare that with permutation, we shuffle that, and this is a result. We can see that the fitness, here's a fitness, is the permutation distribution. If we use only neuronal signal, still there's some information there that we can have a better partition of the cluster, which makes sense because in the beginning I said neurons do contain information. Similarly, the fMRI data is also significant here, but if you look at the neurons with MRI, this is the most striking, strongest result, suggesting that indeed when we make use of both neuronal and functional information, it gives us the best picture. This is the final slide, and we also test that whether we can group them into we can guess whether they're watching the same video or different videos. Here is a p-value, and there are 3 different methods. The line indicating significant value, and you can see in the middle, most of the neurons, they're actually much better than the other 2 methods when we make use of combine both neurons and functional map by fMRI. So here's a concluding slide. So today I have shown you the idea of this mixed selectivity on a single neuron level, and I'll also introduce how these neurons may accumulate information across time with a decoder. This is one thing, but if we look more globally, we can see we can combine the dorsomedial parietal cortex neurons with each of the voxels across the whole brain globally, and then that would help us inform the relationship of how electrical physiology can combine with functional imaging. The key result is that we show with this experience of going to the cinema or video, giving different episodic experiences, like 6 list of videos. And I think they will be the first step for us to understand, to move further to study how episodic memories may actually, how they actually work with this naturalistic sort of setting. So with that, I would like to just thank people who did work. Wang Lei has done most of the physiology, neurophysiology, and Xifeng's analyzing the MRI data. The data was acquired by Michael, who was then in Newcastle. He's now in Germany now, and this is some of my research members. That brings us to the end of this presentation, and thank you for your attention.
¿Alguna pregunta?
question? Yeah, please.
Thank you very much for your talk. It was a great talk. Have you used the mixed PCA analysis? The mixed PCA analysis, in which you can try to decode each of the variables you're estimating?
So PCA is a way to reduce the dimensionality, right? You can have the different components. So we are working using the PCA with this data, like the maps, because we want to see what components that may I think included them on this slide. But yeah, we have the PCA with each of the videos actually. So we're using the map, the neuron pulse signal maps, run down PCA and obtain the components. So we do have that. So one or two issues that I've seen is that even if they have the components, do we know what is inside each of the components? We still don't know, right? We know the variance explained by the components, but it's not as detailed as the features that I have delineated at the display early on. But I think this PCA could be one way to reduce the dimensionality. Yeah. And what about mix? You mentioned it's a mixed PCA. So what does that mix tell?
You try to untangle the contribution of each of your variables to the activity, to the global activity, right? It's a dimensionality reduction technique, like you said. Yeah.
Thanks. Any questions? Yeah. Okay. Thank you very much.